Good morning, students. Today, let me start with this new chapter, Histology of Liver. This liver, it is one of the very important organ and it is the biggest organ present in our body. Some basics of liver. It lies to the right side of body, just under diaphragm. It is the largest internal organ that holds around 13% total blood at any one time. It can store and release blood acting as a reservoir to compensate for smaller changes in blood volume. It uses up to 20% of total energy in the body, and it is made of left and right lobes enclosed by fibrous capsule, that is Gleason's capsule. Apart from these two lobes, it also has two other lobes, that is quadrate lobe and caudate lobe. Each lobe is formed from hexagonal lobules, and there are about 100,000 hexagonal lobules are there in each lobe. And it has dual blood supply, that is receives blood from two blood vessels. Coming to a brief note on anatomy of the liver. The liver is the second largest organ of the body and the largest gland, weighing about 1 to 1.5 kgs. It is situated in the abdominal cavity beneath the diaphragm. Coming to the other features of this liver. As you observe here, this liver has two bigger lobes, that is the right lobe and the left lobe. And apart from this, it also has the other features like a gallbladder, and it has a bile duct. That is gallbladder, it stores the bile juice or hepatic juice, and that flows out of the gallbladder through this hepatic duct. And apart from this liver also has two other lobes, as I mentioned earlier, that is quadrate lobe and caudate lobes, the diagram of which I will show you in the next slide. And this hepatic duct or the bile duct, it enters into the duodenum, but before it enters, it joins with the duct of the pancreas called pancreatic duct, and that forms a very common duct, what is called as hepatopancreatic duct. Vascular supply or the blood supply to the liver. The liver has a dual blood supply, receiving oxygenated blood from the left hepatic artery and the right hepatic artery, that accounts about 25%, and nutrient-rich blood via the portal vein, that is about 75%. Both these vessels enter the liver at the porta hepatis. Coming to the other features of this liver, anatomical features, and almost the same features as it was there in the previous slide. Here also you can make out this largest lobe, that is the right lobe, and the gallbladder here, and then bile duct, hepatic portal vein, hepatic artery, hepatic vein. So these are some other important features that are there in this slide. Gallbladder. It is a small pear-shaped organ situated on the interior aspect of the liver. It is about 10 centimeters in length and 4 centimeters in cross section. Liver is divided anatomically by deep grooves, that is two large lobes, that is the right and left lobes, and two smaller lobes, that is quadrate and caudate lobes. So here in this picture you can make out these other two lobes, but before that let me go through these major lobes, that is right lobe and the left lobe, and between which there is a ligamentous structure here, what is called as falciform ligament, which is the anterior view of the liver. However, the liver, as I said, it also has two lobes, that is a caudate lobe and a quadrate lobe. And here itself you can make out the presence of gallbladder and the hepatic duct. Coming to histology of the liver. So the histological structure of the liver has two major features, stroma and parenchyma. Stroma includes connective tissue, capsule, that is Gleason's capsule, trabeculae and reticular network. Whereas parenchyma includes hepatocytes, blood vessels and bile ducts. This is the section of a hepatic lobule, that is of human, and as you observe here there are a number of lobules can be seen here, and each lobule will have a central large vein, what is called as central vein, and from this central vein there arise radiatingly anastomosing branched cells arrangement, what is called as hepatic cord or the hepatic plates. This is the enlarged view of that section of the liver, and as I mentioned earlier, this is what the central vein, and these are the radiating cords, that is hepatic cords. This is a part of the liver lobule showing the central vein in a very broader manner. And here you can make out the border of this each lobule, okay, and each of this lobule, as I said, it will have a central vein, and from this central vein there arise the radiating cords, that is the cells of cell arrangement of this liver, what is called as hepatic cords. And almost in every corner of these hepatic lobule, this hepatic lobule, as you observe here, it is polygonal shaped, and in the corner it will have a bile duct, it will have a hepatic artery, it will have a portal vein, and this whole area is called portal area. It is called portal area because it has a vein, what is called as portal vein. And some authors, they also call this structure as hepatic triad, as it has three important features like a portal vein, hepatic artery and bile duct. It is referred to as hepatic triad. So this component is referred to as hepatic triad or portal area. The portal canal contains branches of portal vein, as I mentioned earlier, an hepatic artery and interlobular bile duct, that is portal triad. Central vein, in the center of each lobule, as I have shown in the previous slide, it conducts blood from the lobules into hepatic veins. So here in this enlarged view of this liver section, you will observe this hepatic triad here, portal vein, hepatic artery, bile duct, and this whole thing is referred to as portal triad. And here you can make out the presence of this central vein, and this central vein gives rise to these cords, what are called as hepatic cords or hepatic plates. As you observe here, it is clearly shown that hepatic cords are the arrangement of the hepatic cells or the liver cells, which are also called hepatocytes, and usually, as you observe here, each hepatic cord or the plate, it will contain a single row of cells. However, it may show some branches here and there. You see the branching here and one more branching here. So that kind of arrangement of cells will be there. And between these two successive hepatic cords, there is the presence of a space, what is called as hepatic sinusoid or the liver sinusoid. And in this central vein, you can see the presence of these RBCs, and apart from that, you will observe the arrangement of these blood cells within the sinusoids. Okay, so these are some other important features of this liver section. So this is the another diagrammatic view of the liver section, and let me enlarge it or zoom it. So there will be a central vein here, and liver plate, sinusoids, the blue one which is shown in the diagram, the liver sinusoids or hepatic sinusoids, and this is what bile canaliculi, bile duct, portal vein, hepatic artery, that whole thing constitutes the portal triad. So here you can make out these hepatic lobules here. Coming to the details of liver lobule. So each liver lobule, it will have the arrangement of hepatic cells called hepatic cords. So hepatocytes are arranged in one cell thickness and radiating from the central vein, as it is already explained to you, and you can further make out that arrangement here. So this is what the central vein from which there arise the branched anastomosing or the arrangement of these hepatic cords here. And the irregular spaces between the hepatic plates are occupied by liver sinusoids lined by fenestrated endothelial cells. So this is another enlarged view of a single hepatic lobule. So here this is another diagrammatic view of this liver lobule. So it will have the components like portal triad, the label number 1 and 2, lobule, that is each liver lobule, and 3 central vein, 4th one bile canaliculi, and the 5th one common bile duct, 6th one hepatic portal vein, 7th one hepatic artery, 8th one hepatocyte plate or the hepatic cord, and 9th one is sinusoids, that is the space between the two successive hepatic plates or the cords. This is another diagrammatic view of that liver section. So here you can make out the major bile duct, and this bile duct is formed by the fusion of these ductules called bile ductules, and these bile ductules are there between these bile canaliculi. So this is another view of a liver lobule and the blood. And one important thing that should be observed here is always the blood flows from periphery to center. So that is being shown in the blue colored arrow marks here. However, the bile flows from center to periphery. So this is what a very important thing that you have to observe. So this is another diagrammatic view of hepatic cords and liver sinusoids. So as you observe in this slide, there will be the hepatic triad here, which contain a portal vein and a portal vein or venule here, bile ductule and hepatic arteriole. So this whole thing will form the hepatic triad or the portal field or the portal area. And apart from this, the space that is there between these two successive hepatic cords, as I mentioned, it is referred to as sinusoids, that is liver sinusoid. And inside this liver sinusoid, you will observe some stellate shaped cells or characteristic star shaped cells, which are called Kupffer cells, that is von Kupffer cells. And these von Kupffer cells play an important role in phagocytosis, that is the destruction of the foreign substances. And apart from this, you will see the presence of some endothelial cells here that is lining the inner space of these hepatic cords, what is called as sinusoids. And apart from that, it will also have some other stellate shaped or star shaped cells that also play an important role in phagocytosis. And this von Kupffer cells, it is a specialized macrophage cell responsible for phagocytosis. Coming to the sinusoids in detail, hepatocytes have microvilli, that is increased surface area of the liver cells, and it releases plasma proteins like prothrombin, fibrinogen and albumin, and lipoproteins like endocrine functions of the liver and cholesterol. It absorbs insulin, glucose, minerals, vitamins, blood-borne toxins for detoxification. As I said earlier, the von Kupffer cells in the sinusoid, which are phagocytic cells, and these are derived from monocytes, and they ingest bacteria from blood and the breakdown bilirubin for absorption into hepatocytes. Hepatic sinusoids and hepatocyte plates. These are anastomosing plates of hepatocytes. Anastomosing means which are profusely branching and no more than 2 cells thickness prior to the age of 7 years and 1 cell thickness after that age. And the radiating arrangement of these hepatic cords from the central vein towards the periphery of the classical lobule, and the spaces between the plates of hepatocytes are occupied by hepatic sinusoids. And the blood flowing in these wide vessels is prevented from coming in contact with the hepatocytes by the presence of an endothelial lining composed of sinusoid lining cells. Sinusoidal lining cells have gaps of 0.5 microns. Coming to hepatocyte. As I said earlier, the cells of the liver are called as hepatocytes or hepatic cells. And these are large organelle-rich cells that manufacture the exocrine secretion bile as well as a large number of endocrine secretions. In addition, these cells can perform a large array of metabolic functions. And these hepatocytes make up roughly 80% of the mass of the liver, and the nuclei are distinctly round, most single nucleus, and some will have binucleate cells. Hepatocytes are exceptionally active in synthesis of protein and lipids for export, therefore have large quantities of both rough and smooth endoplasmic reticulum and Golgi membranes. This Golgi complex or the Golgi membrane and the smooth and rough ER, that is endoplasmic reticulum, they are involved in the transportation of or the export of the substances between the cells. So apart from that, glycogen granules and vesicles containing very low density lipoproteins which are readily absorbed. Coming to some important functions of liver. First one, carbohydrate metabolism. It turns proteins into glucose, turns triglycerides into glucose, turns excess glucose into glycogen and store in the liver, and turn glycogen back into glucose as needed or when needed. So these are the functions of liver with reference to carbohydrate metabolism. Coming to lipid metabolism, liver synthesizes cholesterol and it synthesizes lipoproteins, that is HDL and LDL. It is used to transport fatty acids in bloodstream and it stores some fat and it breaks down some fatty acids too. So these are the functions of the liver with reference to lipid metabolism. Coming to liver functions with reference to protein metabolism, it brings about deamination, that is the process of removal of amine group from amino acids so that it can be used what is left as energy source. It converts resulting toxic ammonia into urea for excretion by the kidney, and it synthesizes plasma proteins utilized in the clotting mechanism and immune system. It converts one amino acid into another. That means interconversion of amino acids is also an functional attribute of liver. Other liver functions: detoxifies the blood by removing or altering drugs and hormones, that is thyroid and estrogen. It removes the waste product, that is bilirubin. Releases bile salts like sodium taurocholate and sodium glycocholate, which helps the digestion of fats by emulsification. It stores fat soluble vitamins like A, B12, D, E, K. It stores iron and copper, so it is a good storehouse for iron and copper too. And it phagocytizes worn out blood cells and bacteria, activates vitamin D. The skin can also do this with 1 hour of sunlight a week. Coming to liver physiology, it secretes bile, that is a exocrine function of the liver, and it modifies the structure and function of many hormones like thyroxine, growth hormone, insulin, and glucagon. It produces most of the circulating plasma proteins like albumins, VLDL, glycoproteins, and prothrombin and fibrinogen. It stores and converts several vitamins like A, D, and K, and iron. And it degrades drugs and toxins by oxidation and then conjunction. So, this is all about today's class. I hope students you have understood the details of this histology of the liver. Thank you.